Just keep talking.
No, like having like free time.
All right, let's go ahead and get started. So, a couple of quick announcements. First one, the first review went up, and I honestly, it was really, really good. So, to the four of you, great job, honestly.
That was the hardest thing of my life.
It was really, really good, though. Well, I would apologize, but I'm really not going to, because those are your life decisions to go out and drink the night before. So, but really, really good overall. Secondly, we'll keep that up, so check to make sure if you're on this week. I think I actually emailed, did I send an email out? Okay, just checking. So for homework tonight, just so you're aware, or for Wednesday's lecture, it is a group wiki. So what we're going to cover on Wednesday is horizontal gene transfer, which is in contrast to what we've talked about, which is replication is considered vertical gene transfer. So bacterial species have an ability to share genetic information actually within a generation, and so that's what we're going to talk about on Wednesday. What I've put up on the wiki is just a list of terms. I think there's about 20 of them. And what you need to do, each member should take one of the forms of gene transfer. So the options are vertical, transformation, transduction, and conjugation, which are all different forms of horizontal gene transfer. And you need to be able to take the terms that fit with that particular form of gene transfer. So each person takes one out of the list of 20. Just pick the terms that best fit with yours. Some of the terms may fit in more than one category, and that's perfectly fine. But this is a way we'll actually start to mentally organize the information before we talk about it on Wednesday. Okay. And then the other thing, lab on Wednesday. What do you have due? Yeah, annotated bibliography is on Wednesday. Okay, so. Make sure to have those ready to John. I'll create a Dropbox for it. I haven't yet, so you can either bring a hard copy with you, or I'll put a Dropbox in the lab section where you can just drop them in there if you'd like. So one again per group for that one. All right. So what I wanted to start with today was to do a little bit of review from the homework assignment. So each member of the group took one of these different amino acid changes and then identified it as being a certain type of mutation, identified the amino acids that were associated with it, and then finally determining, does this have an effect on the cell or not? Okay, so I wanted to spend a few minutes reviewing this. I'm going to get something, the recording stopped for a second, so let me see if I can get that going again. Blah. All right, so in the meantime, why doesn't everybody take a look at A real quick and share with your partner what you think the right answer is for that one? And I have the code up here too. So A over here, UUA to CUA, is there an amino acid change? What are the amino acids? Use this to help you out. And this may be different than the A that you guys did. I had to it is because I had to change some of these around because I realized after the fact that I didn't give certain ones that I wanted to. So let's just use this as a practice to get us going. Okay. Nope, you did it. You did it correct.
I know.
All right, so UUA encodes what? Leucine, LEU, and then CUA, leucine again. So this is what we would describe as a point mutation, which is just a single nucleotide or single nucleotide change. What kind is this? What kind of point mutation? So is it a missense, a silent, or a nonsense? This is a silent. Perfect. So change in the amino acid, or change in the codon, but no change in the amino acid sequence. Would we predict this would have an effect on the protein? No, it's the same exact amino acid. So if we have a change like this, what we can assume is that the function of the protein before the mutation and after is exactly the same. And the reason why these conditions, these silent mutations can occur is because there's redundancy in the codon, in the codon code. So for example, leucine, there are 6 different codons that all encode for leucine. UUA, UUG, CUU, CUC, CUA, CUG. Okay. What if we went from, I think this problem originally was a UUU or something like that to a UUA. What happens here? Yeah, so we go from phenylalanine, which is PHE, to leucine. What kind of mutation is this one? This is a missense, so change in the amino acid sequence. And then the follow-up question is, does this have an effect on the protein? Or would this have an effect on the active site of the protein? And what would you think? No? Yes? Maybe? Would that mutation from a phenylalanine to a leucine, would that have an effect on the protein? If this were in the active site of the protein where this mutation occurs? Yes? Vincent, you said yes too, right? Why? It could. It could, absolutely. And so sometimes in this case here, now this is going into more depth than what I was necessarily asking for in the homework, but it can change the folding. So sometimes we actually, in these missense mutations, have to take a look at, well, what do these amino acids look like actually? And this is not something that I would ask you to memorize. Did you have to memorize these in biochem? Yeah, that was a horrible experience, and so I have my cheat sheet here today to remind you. Allie.
What do you mean by active site? Like, the site that's like actually...
So the active site, I would mean like if this were an enzyme being mutated, the active site is where the substrate binds.
Yeah.
So if for example phenylalanine were an amino acid within the active site, because it's amino acids that make up that active site of the protein. If you mutated that phenylalanine to a leucine, would that then change the active site in a way so that the enzyme can no longer perform its function? Or does it less efficiently? It could make it less efficient, yes. How do you know? You usually don't know unless you actually test it. So that's not something we would necessarily be able to know with certainty just by looking at it, but we can make a prediction based on off of what we observe. So let me give you an example here. In this one, I'm just going to draw, so here's our carboxyl group of an amino acid. There's that hydrogen. Here's our amino group. This particular one has the following. Has a 6-carbon ring, all double bonds. Okay, so that's what phenylalanine looks like. Leucine has the same basic structure as do all amino acids. But the difference with leucine, there we go. Wait, I didn't do that right. Oh, damn. Bother. Don't erase the whole thing. Okay, there's leucine. So we would look at this and then say, what are the properties of these two? And are they similar in size? Those would be the most immediate questions we might ask. So, properties of this, which is made up of all carbons and hydrogens? Hydrophilic, hydrophobic, polar, nonpolar? What might we assume, given what we observe? And let's just talk about the side chain. Don't worry about all of this up here. Just focus on these fragments here. It's going back all the way to week 1. Told you this stuff would be important. Cassidy, do you have a question? It would be hydrophobic. Hydrocarbons, hydrogen and carbon, are nonpolar bonds. Nonpolar bonds lead to hydrophobic properties. So we might assume that, yeah, they'd act similarly because they're both hydrophobic side chains or both hydrophobic amino acids. You might also say that, well, this one is different in shape. It's much larger than this is, and so that might actually affect the function. So you can at least make a hypothesis or a prediction based on what you observe. Okay, but you wouldn't know for certain unless you actually tested it. Does that make sense, Allie?
So like on the exam, there's a question you really draw that.
Oh yeah, I'd absolutely give these to you. I would not just ask you to figure it out on your own. No, that, like I said, this is one of those things that I don't really remember what they look like, but yeah, so definitely let's do that. Let's take a look at another one real quick. I'll give another one where we might predict there to be a similar function. So, erase that real quick. If we went from, I think one of the ones was like nope, that's not right. AUU to GUU. What is this particular change? So it goes from what to what? AUU encodes isoleucine ILE. GUU, val, valine. So these two, if we just draw, again, I'm just going to draw this as the basic structure. That's isoleucine. Here's valine. So similar in structure, different in structure. There's a little bit of difference, but overall it's pretty similar. Both of them have carbons and hydrogens, so we'd assume that they're hydrophobic. There's only single bonds in here. Those are movable. The only difference is this isoleucine has this extra CH3 group right here. And so we might assume in a case like this that because there's a similarity in structure, it retains the same function. Maybe there's just a little bit of loss of function. So we can at least make predictions based off of this. And the reason why I want to point this out is not so much to get you to freak out that, oh my gosh, he's going to force us to do all this. The reason why I want you to think about this is oftentimes we think of mutations as being a really bad thing. We often think of them as being a negative effect on a cell. And sometimes they can be beneficial, even in cases where they're missense, like this one. They may have very little effect overall on the function of the protein because the structure remains largely the same. Okay? So I wanted to just point that out. What's the last type of mutation that we haven't talked about yet today? So we've got silent, missense, and what's our last one? Nonsense. And what's the hallmark of a nonsense mutation? Yeah, so the code changes from an amino acid to now encoding for a stop codon. And those definitely are going to have an effect. Obviously, if you have a protein that's 200 amino acids long and translation stops at amino acid 100, well, you're missing the whole other half of the protein sequence, the amino acid sequence, that would contribute to the overall structure. Okay, so those are definitely going to have, we can assume, a negative effect on the cell. Any questions on these particular mutations? How do mutations form then? In what ways can mutations form in the DNA that lead to changes in protein structure. I'll give you a minute to talk with somebody else about them because I can see we're a lively group today. So turn to a partner next to you and tell them, hey, mutations can occur by fill in the blank. We'll play a game, a match game. All right, so how can mutations occur? Radiation, both forms, ionizing and UV. So in the case of ionizing radiation, this creates double-strand breaks. In other words, those sugar phosphate backbones get broken, essentially. And in the cases where that occurs, if there's enough ionizing radiation, we talked about how this is an incredibly effective growth control mechanism. Bacterial cell basically halts in its division when that occurs. Because oftentimes, in cases where we get this stop because of ionizing radiation, let's assume that this is our bacterial chromosome. Those double-stranded breaks are going to occur and create just basically flat ends. So we'll get a bunch of short fragments. Now, try putting together a puzzle together that has no little grooves or anything. It's just a bunch of flat little pieces. And that becomes a pretty challenging puzzle to put together. And that's what bacterial cells are trying to do here. It's not like there's overlapping sequence so that they can perform some base pairing or complementary base pairing. In essence, they've got to try to piece this back together. And oftentimes what this will lead to are mutations, lost DNA, sometimes duplications in the genome. Overall, going to lead to stopping the cell from replicating. UV radiation, on the other hand, creates thymine dimers. So UV radiation, when it hits two thymines that are next to each other in a DNA sequence, it causes them to form a base pair with each other. Or not base pair, sorry. Causes them to actually covalently bond together. And so the result then is a little pucker in the DNA, where they have now bonded together. And that can lead to a misreading of the DNA then by DNA polymerase. And so it can lead to insertions, deletions, maybe just a point mutation as well. And there are other mechanisms that involve chemicals. So what we describe as being carcinogens, things that cause cancer, they are chemicals that increase the rate of mutation by affecting the structure of DNA in some way so that it just increases the rate of mistakes that are made by DNA polymerase. One other way, as Zach pointed out, is randomness. Random or spontaneous mutations that occur just as products of mistakes from the DNA polymerase. So roughly the E. coli DNA polymerase, even after proofreading, makes about one mistake every 10 to the 9th base pairs, roughly. That means that for every 220 to 2200 cells that are generations, there's going to be one cell that's different than the rest of them. In other words, in the cultures that we grow up in the lab, oftentimes we think of the cells in there as being identical to each other in terms of their sequence. They're actually different, though. There are some cells that might have a spontaneous mutation that has maybe led to the mutation of a gene. And that mutation could be a silent mutation, could be a missense mutation, could be nonsense, may not even be in a gene encoding region of the genome. We can't necessarily tell unless we try to tease that mutation out with some sort of selective advantage. Okay? And so this is where we're going to move into talking about Neisseria, because Neisseria has been this really interesting model, specifically over the past 20 years, 20 to 25 years, and looking at how selective pressures create a population of cells that are antibiotic resistant. Okay? Any questions on how mutations occur? Okay, so what I'm going to do is I'm going to flip to a different set of slides. I posted these earlier today. You did not need to print them out. And we'll come back to this a couple of times later on. But this is where the worksheet comes into play. So what I did with the worksheet there is a couple of things. First off, what I've done is the first 4 questions are kind of follow along with what I'm talking about. And then we'll stop and we'll work on the 5th one for a little while together just to talk some more about these mutations the other day. But what we get from this process then are, if this is our typical bacterial cell with a chromosome, when we add a drug like ciprofloxacin, cells continue to elongate. It's got 2 chromosomes, but those 2 chromosomes don't separate from each other. And the cells basically kind of freeze or halt in this point of their division from each other. So we would say that ciprofloxacin is bacteriostatic. It inhibits the growth of the microorganism. Isn't necessarily bactericidal within 24 hours. So here's what's happened over the past, from 1990 to 2005. This experiment is looking at the percentage of Neisseria gonorrhea isolates, clinical isolates, that were either considered to be resistant or considered to be intermediate to ciprofloxacin. Given that we're measuring resistance and intermediate, what kind of experiment is being performed here to determine this designation? So is this a minimal inhibitory concentration or a Kirby-Bauer assay? It's a Kirby-Bauer. The Kirby-Bauer is the one, and we'll take a look at this on Wednesday in lab, that's actually associated where at the end we can list microorganisms as being resistant, intermediate, or sensitive to these various drugs. So track from 1990 to 2005, what happens? What's the trend indicating? Increase in what? Yeah, increase at least in the number of isolates that are found to be resistant, right? So we go from basically maybe less than 1% that are intermediate. That maybe increases to like 2% or so by the end, and that stays pretty consistent. But the percentage that are identified that are resistant is almost about 10%, 9 to 10%, 1 out of every 10 or 11 isolates resistant to ciprofloxacin. Okay. So how does this occur? One of the ways that we can look at this is by going back to, if we know the target of ciprofloxacin, which is gyrase, we can actually start to look at the DNA sequence or the amino acid sequence and identify, are there particular mutations that correlate with these phenotypic changes, this resistance to an antibiotic. So this is a table looking at 6 different strains. So FA19 is considered here the wild type or the original strain. And then A, B, C, D, and E are all strains that have increased resistance to ciprofloxacin. These are MIC values, so minimal inhibitory concentrations. So they are ordered from lowest MIC to highest MIC, and as that MIC increases, we say that the organism is more resistant, takes more drug to inhibit its growth. These organisms are also resistant to nalidixic acid, which is another quinolone, so it's in the same family of antibiotics as ciprofloxacin. How about these two, novobiocin and penicillin? Is there any effect? And the answer is no. It does not seem that these isolates are changing in their sensitivity or their resistance to novobiocin, which is an aminoglycoside, so inhibits the translational machinery, or to penicillin, which is a beta-lactam, so inhibits cell wall synthesis. Okay. So let's start here, and this is then question 5 on that worksheet. What I want you guys to do is we're actually going to work backwards now. We've worked forwards in looking at how does a change in amino acid sequence affect how does a change in the codon affect the amino acid sequence? We're going to work backwards from what we know about the codon code and figure out what was the specific nucleotide change that occurred that has led to these changes in resistance to these antibiotics. So let me just orient you with this table real quick. 83 to 104 here represents just a 20 amino acid range, or 22 amino acid range in the gyrase gene or gyrase protein. And what you should pay attention to here is that at amino acid 91, there was a change from a serine to a phenylalanine in 4 of these different mutant strains. And at 95, there was a change from aspartate, ASP, to asparagine, ASN, in 3 of the strains. And you can see how these changes here correlate with increased minimal inhibitory concentrations or increased resistance to these antibiotics. What I want you to do is to walk through this. Identify what are all the potential codons for serine, what are all the potential codons for phenylalanine, and then identify how could those mutations have occurred, and identify what nucleotide is most likely mutated, okay, to lead to those changes. I'm going to give you about 5 minutes to work on this for both of these. If you fly through them, continue on in the worksheet. There's another set of mutations that correlates with this resistance as well. And we'll take a few minutes to talk about it after you finish up. All right, go ahead. Okay, so what's 6? I think there's 6 for serine, right? UCU, UCA, UCG, UCC, and then what other 2? AGU, AGC. And then what for phenylalanine? What are 2 for phenylalanine? UUU, UUC. So given what we have here, what potential mutations could have occurred leading to a change from serine to phenylalanine? So UCU to UUU, good. UCC to UUC. In both of those cases, what nucleotide has changed? Yeah, it's the second in that codon. So basically, by going and doing this reversed method, we've just identified the single mutation that's occurred in these strains, which has increased resistance to ciprofloxacin. Okay, and to point out here, serine is a polar. It's got a hydroxyl group on it, so it's a polar amino acid. And phenylalanine, we've already taken a look at. It's hydrophobic. So there's a difference in property there that could explain the inability of ciprofloxacin to have an effect. Most likely, it's affecting the ability of ciprofloxacin to bind to the antibiotic, because we've talked about how a lot of these antibiotics are competitive inhibitors, that they're competing for the active site of these particular enzymes. How about the next one? So aspartate, which is ASP up on the right codon code over there, and then ASN is asparagine. So what are the codons for aspartate, ASP? GAU and GAC. And for asparagine? AAC. So in this case, Mutation is? Specific one is that G to A, again, changing the qualities of the amino acid. This one is a little bit different. Aspartate has a negative charge at neutral pH. Asparagine has a positive charge at neutral pH. Both of them are polar, but the change in the charge there seems to have an effect as well. Okay? Sid?
How did you figure out the difference in ASP and ASN?
This 95 there, it lists D as being the aspartate, and then the N below it indicates that there's an asparagine there.
How do you know?
How do I know that?
So do you use
Yeah, D is ASP, sorry, yes. Nope, nope, I would give this information to you. And then N is ASN. Yeah, absolutely. Questions on how to perform this. So this is just a different way of looking at it. This is looking, starting at the end, how can you figure out the specific nucleotide change? Whereas before, for homework, we were looking at how does that nucleotide change affect the amino acid sequence. It's just a slightly different way of actually looking at this. Any questions with that, though? What kind of mutations are these? What kind of point mutations? Yeah, these are missense, both changing the amino acid sequence. I want to just point out here that with the worksheet, then the rest of the questions are kind of a follow-up. So this wasn't the only mutation that they observed in these strains. For example, you can see these bottom three here, C, D, and E. They have quite varied difference in their minimal inhibitory concentrations for ciprofloxacin, but they all have the same 2 amino acid changes in gyrase. They ended up mapping the last couple of mutations to actually PAR-C, and that protein encodes topoisomerase. And topoisomerase is basically redundant in function with gyrase. Both of them are involved in reducing the supercoiling. And what you see here is that as this resistance increases, there's actually an increase in the number of mutations at these sites where ciprofloxacin would bind. So the one that had just one mutation, there's the MYC is 0.25. The second one had 2 mutations in gyrase A, MYC is 0.5. Now if we get mutation in both PAR-C and gyrase, now there's quite a dramatic increase in that minimal inhibitory concentration. Go from below 1 to 4 or 16 as the number of those mutations increases. Okay. In essence, what happens in these cases here where we find these mutations occurring, I kind of tried to model this as best as I could. But what happens is, if we think of these antibiotics as competitive inhibitors, the structure of the protein changes enough so that that competitive inhibitor can no longer bind, but the enzyme is still able to perform its function because it's still retained enough of its 3-dimensional shape. Oftentimes there's a bit of a trade-off here. That trade-off is, for that resistance, the enzyme oftentimes will lose some of its functionality. So for example, gyrase might not, this mutated form might not as quickly be able to reduce supercoiling, or as quickly be able to separate those 2 strands of chromosomes from each other, which would just reduce the rate at which cell or binary fission can occur, so increase that generation time. So I just wanted to make sure to point that out real quick. Any questions on this concept of antibiotic resistance? So again, this goes back to specifically, if we're taking a look at those 4 forms of resistance, this one goes back to changes in the target. Changes in the target that lead to resistance. Any questions? Okay. I'm going to hand back exams, and overall, much improved over the first one. The average score in class went, on the first exam it was right around a 74. This time it went up to an 81. So it's almost a 7-point gain just right there, which I was really happy with. The most common issues were a couple of the essay questions, and I would say maybe the one that I saw most frequently being an issue was actually the last question that asked about between rifamycin or tetracycline, which one would you choose to treat this infection? And it came back to calculating the therapeutic index, which a lot of people did do. If most people noted that there was a difference in that rifamycin worked better than tetracycline, and that probably correlated to therapeutic dosage, and that was correct. The toxic doses were actually listed in the test as well. It was in one of the tables that listed all the antibiotics. And so that was kind of what I was looking for, was an explanation of overall the therapeutic index is greater for rifamycin than it is for tetracycline. So that was probably one of the most common issues. What I would say is, again, take a look at these, take a look at what's been posted for the key, and then when you are ready, come by and talk to me if you'd like to about the exam itself, okay? As always, check the points, make sure I've added correctly. The in-class portion was out of 77 originally. Somehow I screwed up on how many points I made it. So I just multiplied your score, or divided it by 77 to get a percentage, and multiplied by 81, because that's how many I was trying to make it out of. So if you have questions about that math, let me know as well. But other than that, once I hand these back, you guys are free to go, and I'll see you all on Wednesday.